Now, I want to welcome Dr. Carman Giacomantonio, our principal investigator on the study, to share a few more thoughts here. Carman, you're sideways and you're labeled as David Regan, but that's fine. There we go.
There you go.
My twin brother clone. If only it were so. Here's Carman.
Our parents are not very happy with this designation, so well, at least yours. So thanks, David. So and Len. So essentially, what we are hearing is that this gold nanoparticle is being used initially as essentially a tumor destruction agent or a primary tumor treatment agent. And what we're branching into with this discussion is it as an adjunct to an immunotherapy or even an adjuvant to immunotherapy. Next slide. Yeah. So as Len mentioned, we know that cancer cells are particularly sensitive to thermal energy. Cancer cells thermal energy essentially drives a cancer cell into a death cycle that normal cells are generally resilient to within a range of temperature. One of the things that's particularly important about the gold nanoparticle is we can control the amount of energy that's generated. What happens is, when we inject the particle either intravenously or directly into the tumor, we can activate it specifically where it lies. The targeting is within centimeters of the energy source and limited to activation just of the particles that experience the energy from the near infrared light. This is very important because many of the other targeted therapies that we currently struggle with in health have more global effects, either systemic immunotherapy, systemic chemotherapy, or radiation. And radiation has, as many of you would know, a collateral damage challenge that we're left with dealing with the aftermath, both in the either the surgical setting or in the healing setting. So immunotherapy, we know, is currently leading all cancer therapies, and it's been superbly effective in some cancers, yet in other cancers it's had limited efficacy. And there's lots of theories to why it would have an ability to treat a melanoma quite effectively, but not treat a breast cancer effectively. Probably the most primary reason that this doesn't happen is the antigens that are generated in these cancers, the signals that excite or interest your immune system are just not apparent. The gold nanoparticle promises to enhance that by creating local tumor destruction, and with local tumor destruction, as a part of the process of cleaning up the normal biological process of cleaning up the tumor necrosis, the dead tumor, is the presentation of some of that tumor to the immune system. We have seen this in other kinds of cancer therapy, radiation therapy, some toxic therapies, that when that happens, serendipitously, the immune system actually recognizes the cancer in a new way and takes over and kills the tumor. This is the strategy that we're focused on here. Next slide. So what happens? So we're in the early phases, and what will success bring us from the lab? So the first thing is that it'll confirm that we can kill the tumors, we can shrink the tumors, we can cause the necrosis that we've been talking about in a controlled fashion in the mouse model. We're going to try this in breast cancer, melanoma, and colon cancer. The second thing is we're hoping that we can enhance the immune experience. And so what this means is this: we anticipate, with good evidence to support this hypothesis, that the thermal destruction of the tumor will generate the antigen that we spoke of. And this, in combination with some local immune therapies, should create the immune-directed response that we believe is at the core of this tumor killing. So what I'm saying is that although the thermal energy will drive the cell into a death cycle, the actual clearance of the tumor is almost assuredly an innate immune response. So the final thing is, next portion, is that there is a reasonable anticipation that at least in some of these animals and some of these experiments, we'll see a response that goes beyond the primary tumor. Why do we expect that? Because tumors, as you know, are associated with not only local growth, but developing of metastasis. Those metastases or daughter cells or offspring of the tumor will very often look exactly like the primary tumor. So when you generate an immune response to that primary tumor, given that the immune response is truly a systemic response in that the cells that are released have to circulate and come back to the tumor that's been treated, any cell expressing a tumor antigen like the one that's been primarily responsible for the response would be attacked by the immune system. This is what we call an abscopal response. Now, this is a hopeful this is a hopeful response at this point. We do see this in clinic from time to time in the context of radiation therapy. We believe our hypothesis is correct, is that we're going to see this in the animals, and we've designed the experiments to test this hypothesis. So what are the implications for all of this? What are the things? Well, the first thing is we get the experiments done. We'll prove the primary hypothesis is that we can kill tumors locally. We can generate some kind of immunity, and hopefully that we can reproduce. This is a reproducible phenomenon. In the context of the science world, this will allow us to present this data at local, national, international meetings. And often at these meetings, and almost invariably at these meetings, is where the potential partnerships would be struck to enable us to talk to other scientists, to look at partnerships in other labs, partnerships with industry who are constantly combing these scientific meetings to see where the next where their next opportunities are to not only to learn new things, but to add on to the strategies that they're already using. And this is clearly where this will ultimately go for us. Next slide. So the enhanced immune response that I'm talking about, that I'm really excited about in terms of this work, is demonstrating that the immune pathway that we've established is, or the killing pathway we established is immune-based. And then presenting the strong evidence from our research to other labs that may want to collaborate, and to other companies that may want to support or even enhance what we're doing, showing them that the scientific pathways, the biological pathways that we have established are reproducible, they're understandable, they're not serendipitous, but they're intentional.
And that's been the challenge to date, right? I mean, it shows up occasionally, but it's a question of making it.
Well, this is the big challenge with abscopal responses for sure, is they're inconsistent. We see them, and we know that these responses happen, and for the most part, many of them are phenomenal. Even to this date, when we use current immunotherapies, we know the immunotherapy is working because it's enabling the immune system to continue to do what the immune system does. But we know with current therapies that without an established immune relationship, the immunotherapy that we give won't work. It won't enhance what doesn't exist. The therapy that we're talking about with the immunity initiated through tumor destruction from the thermal energy of the nanoparticle is new in immunity. And so in the context of the harmony of doing this with the immune system, our hypothesis is that we're going to generate new antigen that's tumor specific, that is systemic. That's the abscopal response. We need to take it from phenomenon to process.
That's fair. Would you say, we've got a question here, but asking if whether the combination of targeted hyperthermia therapy, our tumor destruction therapy, together with an immunotherapy, would result in an easier path through clinical trials and approvals?
Well, that's a great question, and the short answer is it's a logically easier path because we have current immunotherapies that have already been rigorously tested in the clinical setting, and so the safety and efficacy is understood, and so are the limitations. And so clearly, if we combine that with the gold nanoparticle, which which we anticipate will be very well tolerated in the clinical setting with minimal toxicity. These two, this is the natural next step, and it's the easier path because again, if my contention is correct that the gold nanoparticle resulting tumor destruction is ultimately an immunological pathway, then certainly the checkpoint inhibitors will advantage the immunology, because if the antigen that's exposed is yet again a weaker antigen, using the checkpoint to address that concern should enhance the immunity. That's the right hypothesis, and it is the natural pathway for this to move as the kind of low-hanging fruit, if you will. The finesse, the elegant science beyond this will be combining the two within the tumors to more critically analyze the tumor microenvironment, the antigens, the T cell receptors, et cetera. That will be a part of the tomorrow's research.
Excellent. Thank you for that, Carmen. I don't know if there's anything more, but if you could stick around for our Q&A, we'll have that in a few more minutes. And,
Sure.
and certainly it is our expectation that, yes, as you put it well, it's a logical path forward to see existing products in market looking to have enhancements to those products and extensions to their life by adding on capabilities such as the ones we're exploring right now.
Exactly.